David Varon to talk about platelet microparticles in health and disease.
So, good afternoon. Many thanks for the organizers for the invitation. This is my first time, my first brief visit of this wonderful place, to be continued later, even in the summer. And I will keep the momentum that Lina started by talking about non-hemostatic functions of hemostatic parameters. And I'm going to talk surprisingly on platelets, and I will be talking on platelets and microparticles mostly, and I will give you an overview about different functions in this field of inflammation, immune responses, angiogenesis, tissue regeneration, and cancer progression. And it might not be that much of a surprise to realize that platelets are capable of doing many other things except of hemostasis. When we observe that in this ancient creature, billions or millions of years ago, this is a horseshoe crab, Limulus, which could still be found at the Cape Cod shore, has only one cell in its bloodstream. So this cell can do everything. It is like erythrocyte and leukocytes and thrombocytes. So they still remember. Okay. So it's not a surprise, and we know for a while that platelets are equipped with many of the players, at least of angiogenesis. This is just an illustration of a partial list of all the components that they have. And it is just the question, and if you can see these, all of these green or sort of green, yellow are pro-angiogenic, and the red are anti-angiogenic, and they have many other components. And the question is how this is operated, and is it controlled by any mechanism? A little, 2007, Italiano and his group were trying to solve this issue by demonstrating that different triggers or different stimulators of power 1 and power 4 induce different release of these agonists, of this cytokine, and they even were able to show a differential distribution of the cytokines in specific granules. So it is not quite clear by today that this is the whole story, but at least it shows that within the platelet granules, there is some distribution, differential distribution of the different cytokines, and stimulation of release from one would create increase in VEGF, and the other one will create really increase in thrombospondin, for example. No, that was... And to show something in addition, and this comes from Jalnula Clement, who was born here, but now lives in Boston. And they show that platelets do not only get their granule content from the megakaryocytes, but they can also acquire them actively from the cytoplasm. And for example, cancer patients show that an increase in the VEGF concentration in platelets as compared to the plasma, as compared to normal platelets. So granules of platelets from cancer patients are exhibiting an increased concentrations of cytokines. This is another way by which platelets acquire this agonist. And last but not least, introduction, platelets were found also to be a mirror of the cancer situation. Here, a work by Woedinger from Holland, showing the RNA signature of platelets from cancer patients versus healthy volunteers. So you can differentiate, this is a wide range of RNA evaluation, and you can see a strict differentiation between healthy and patients with different types of cancer. In other words, platelets contain RNA which in cancer patients, there is some types of RNA that do not, they are not existing in healthy volunteers. Now, let's talk about inflammation a little bit. This is work by Tim Springer quite some time ago, showing that platelets are very actively playing a role in homing and adhesion of neutrophils and penetration of neutrophils into the inflammatory region. So platelets contain a mechanism that allow leukocytes, in this case granulocytes, to roll, adhere, and penetrate into the inflammatory site. This is one example. Here we can see that activated platelet, this is something more recent publication, activated platelet release extracellular mitochondria. And these mitochondria may affect neutrophil, and they are inhibiting, they are inducing pro-inflammatory mediators. So again, platelets secrete mitochondria, neutrophils are affected, and secrete pro-inflammatory cytokines. It reminds me of a new treatment, experimental treatment in ITP, in which by low intensity laser, the mitochondria is recharged by energy and increased platelet count. This is something that I just heard recently. Another encouraging work by Eric Boillard from Montreal show very nicely that platelets amplify inflammation in arthritis via collagen-dependent microparticles production. I don't have time to go through these details, but this finding demonstrates previously unprecedented role of platelets as a mediator of inflammation by inducing microparticles that are inducing this arthritis. Okay? And recent work showing also in the inflammatory section that microparticles produced by activated platelets carry patent and functionally active angiogenic signal in Crohn's disease. So inflammatory state, platelets in this case are producing pro-inflammatory signals, and it is always the question of the egg and the chicken, what comes first. And we definitely should look for potential effect, the reverse side. How about immune response? Here is a very interesting and good work by the group of Steve Watson from Birmingham, showing that amplification of bacteria-induced platelet activation is triggered by Fc gamma R2 receptor, integrin alpha-2 beta-3, and platelet factor 4. And in other words, these mediators are involved in the amplification of bacteria-induced platelet activation. We knew for many years that bacteria are, have crosstalk with platelets. Here we show, they show the mechanism, the potential mechanism by activating the gamma FcR2a receptor. And the conclusion of this study was that Fc gamma RA2 mediated activation is common response mechanism used against a wide range of bacteria, and the release of secondary mediators, fibrinogen, platelet factor 4, serves as a possible positive feedback mechanism activation through the IgG-dependent pathway. And how does it work? The Fc gamma receptor is attacked or attached by the anti-platelet, for example, or by a heparin-induced antibody, you know, the platelet, heparin and platelet factor 4. And when you have a cluster of this interaction, or just by a cluster of IgG, and this leads to activation of the cell, clustering of those few Fc gamma receptor induce activation. And this is the way it works. Another important work by the group of Greinacher from Germany, they examined the presence of heparin-induced thrombocytopenia antibody without exposure to heparin. And they showed that here is what the bottom line of the study. They showed that the severity of periodontitis, people with periodontitis, the higher was the severity of the disease, the higher was the titer of the IgG, IgM, and IgA anti-heparin platelet factor 4 heparin antibodies. In other words, these people never saw heparin, and they still produce these antibodies to show that the feature of heparin-induced thrombocytopenia links to a natural effect of platelets as an innate immunity system, because these platelet factor 4 serve as a bridge to other antigen expose and induce antibody production. Let's move now to the microparticles, and we know that they are involved in a variety of mechanism and pathophysiological conditions. Just to show a brief increase in the interest from the 70s till quite a few years ago, you see the amount of publications which is going now. It should be much, much higher. Now they call microvesicle. We studied effect of platelet microparticles by producing microparticles by two different methods. The first is simple activation of platelets by thrombin, and the second is by shearing platelets under a matrix that are inducing platelet activation, and then collecting the microparticles from the supernatant. Using these two methods, we observed different constituents of proteins. This is proteomics analysis, and we found quite different proteins in the microparticles produced by activation by thrombin versus those which were produced by the shearing system. And this is how they look. This is a cryo-electron tomography of a normal microparticle. You can observe all this is the membrane, the 2B3A receptor, and the cytoskeleton. And this is, by the way, microparticles from Glanzmann thrombasthenia patients, platelet, and you can see very little, if at all, integrins on the membrane, and the cytoskeleton is also very limited and not organized. Interestingly, you can use this method to observe the state of activation of the alpha-2 b beta-3 integrin, non-active and active accordingly. So what can we do about, how do we learn about angiogenesis? We showed that earlier that platelets as a cellular system exert a pro-angiogenic effect through potent inhibitors, although potent inhibitors of angiogenesis like platelet factor 4 and thrombospondin are also present in the platelets. The effect, all stages of angiogenesis, and the changes of the amount and the functional state of the platelet may influence angiogenic-dependent processes. And I already told you about the different differential release of granules by different agonists. And as Lina already showed, this is Olga in the group that showed that microparticles induce sprouting of aortic ring micro vesicles, if you want. These are not arteries. And you could block it by VEGF, by FGF antibodies, and by PDGF, and not so much by heparinase. And it was quantitated, obviously. And also, she went later on to see if that has anything relevant in clinical or in biological setting. And here we see vessels of myocardium, healthy, normal myocardium, when you induced a myocardial infarction in a mouse model, and you check for these these vessels, you see very little new vessels at the area of the peri-infarction area of the myocardium. This is an experimental method. If you, at the time of the induction of the acute coronary syndrome, you inject into the muscle of the dependent area, you inject microparticles, you get an increase, not normal, but an increase new vessel formation as compared to the control, untreated control. We did similar studies on ischemic stroke model. This is rats. And Yael from the group tested the effect of injection of microparticles to the lateral ventricle on the same side of the induction of the stroke. And you could see that according to the, this is the score of the score of the functional score of the rats. This is the beginning. They are all healthy. The one who were not treated or by sham injection reached some improvement, but very little. Those that were treated by high dose of the microparticles almost recovered from the deficiency in behavioral score after 90 days, as you can see here. And these are also showing the effect of promotion of neuronal cell and astrocytes by this method. And let me just move to the last part of my presentation, and this is the bad news. The good news were related to angiogenesis and tissue regeneration, and the bad news, obviously, is related to cancer. So the first hint for relationship between platelets and cancer came from Gasik in his group in the middle of the last century, and they showed a correlation between number of platelets and number of metastases after injecting IV carcinoma cell line. This was tested in the lung. So the platelets were decreased artificially by antibodies or by chymotrypsin to make them not functional. So platelets were shown for the first time at that time to be involved in cancer progression. And skip this. And Judah Folkman was one of the initiators of this field. He died more than 10 years ago. And that was a hypothesis later to Lancet that he published with his colleagues. We were able to observe some time ago that platelets mediate tumor adhesion to endothelium under flow conditions. This is what we could see. These were all melanoma cell lines. This is the endothelium. If you do not mix the tumor with platelets, they do not adhere almost. And here you can see adhesion that was found to be significant. And later on, we were able to show that now microparticles of platelets were adhering to prostate cell carcinoma. Here, these are the microparticles. And then if you took the cells after the incubation, you see that they acquired features or receptors of platelets. This is 2B3A. Okay, so these cells became equipped with adhesive protein that they do not have originally. And this is not showing the video, but if it would work, you would see that after incubation of this carcinoma cell with microparticles, these carcinoma cells are flowing much slower than the one that were not treated, that were just rushing in vivo in a mouse model. And also, we were interested to investigate the way this effect is manifested, and we look for the invasion of tumor cells. And here you can see that this is the negative control after 5 hours, a very little invasion. And this is when you mix the tumor cells with microparticles, you see a very significant increase in the invasion of the tumor cells through a matrigel. And we looked for the potential players that play a role in this. And we saw a number of that was a gene array evaluation. So some of the cytokines were upregulated, and some of them were downregulated by the introduction of the tumor cells to the microparticles. And the final was that the effect, the bottom line was the platelet microparticles induced a more aggressive type of cancer with more features of higher metastatic feature. And we were trying to look for the potential players, what was the mediator, and the final suggested member was the angiopoietin inhibition, which was found to be the one that linked to this effect of the microparticles. And also, Olga was able to show increased metastasis of lung cancer in a model of mouse once these cells were pre-incubated with microparticles. We'll skip this one. But just to make the story not so bright and not so easy, a recent finding by another group published in Blood a couple of years ago suggests that I told you that platelets are containing RNA, different RNA once they are exposed to tumor. Here, this group shows that platelet microparticles infiltrating solid tumors transfer microRNAs that suppress tumor growth. So the short description of this is that microparticles containing this miR-24 are inducing tumor regression. So there are also effects that are pro-tumor progression and not only or against tumor progression. So I think that with this I can make a final conclusion and hope I've convinced you that these Miss Platelet multitask are capable of doing good things, hemostasis, immune response, tissue regeneration, but also thrombosis, inflammation, and cancer progression. And with this, I wish to thank all these people that were involved. And I am inviting all of you to the next Platelet meeting in Boston next year. Thank you.
Thank you. Presentation. It's open for questions right now. In the meantime, let me start. I mean, platelets are very difficult organelles to work with because, as you said, they acquire compounds from the circulation, but also when you get them out of the circulation, depending on how you get them out, they can change function. I mean, if you get them in citrate, in EDTA, or if you share them, because when you share blood, you have to have an anticoagulant to be able to share them, right? So do you think that this can produce the changes you saw in proteomics? Because, I mean, the feeling is that you have microvesicles derived from platelets that are so diverse, with so many compounds. Could also be that we are getting them different. They are not always the same, and they are different in the components they have. And really, we will require to classify them very well before we can ascribe functions.
Yeah, this is quite sure. And we actually found by proteomic approach, differential expression of different proteins between those two systems or methods of production of the microparticles. So definitely, and all these exosomes now, we're talking about microparticles and exosomes and endosomes and so forth, are still not yet very systematically being defined. And the field is still, there is now a society which is organizing meetings and publishing. There is also also papers. They produce. They have their own journal. Yes.
But platelets have an additional complexity. We try to do microparticles from platelets, stimulating with thrombin and also with my chamber, as the same approach. And in reality, the content was so diverse that coming from the same donor, that we figure that maybe when we are anticoagulating them, we are already producing a change because of calcium dependent, that then we measure different things. Obviously, this doesn't have to stop us of research, but nevertheless, it's very complicated, more than endothelial cells or another cell, you know, because they are so difficult to get out of the blood.
Endothelial cell. Well, but now when you can grow platelets in the petri dish, that might be, and this is going now on. There are several groups that are, including commercially, trying to make it possible, but forget about commercially. You can take those platelets, but who knows whether these platelets are going to be the real ones.
The same, yes.
Yeah.
You have a question? No? Is
Yes,
any?
if I want
Yeah.
the others, there's nothing?
No. Okay. George?
Thank you. But congratulations, Professor Varon, but I have a question for you. Just a short question. It's for me of clinical importance because you said, if I understood, that we may have heparin-induced thrombocytopenia in the absence of previous exposure to heparin. Is it correct?
I didn't say we have heparin-induced thrombocytopenia. I said
HIT.
HIT antibodies Now they call it autoimmune heat, not heparin-induced heat. It's not heat because there is no heparin there. But what I was trying to make, the point I was trying to make is that different stimulus induce production of antibodies that together with platelet factor 4 are attacking bacteria, attacking invaders. And that's, as I said, and by the way, the bacteria are equipped with sialic residues, which are sugars, which are very similar to heparin. And it could be also the endothelial cell. So the heat is just a hint, if you want. It's like a hit and hint for what platelets can do as they still remember as part of the innate immunity system. And also, it hints also for the possibility to approach such clinical conditions by a microbiome approach.
Thank you.
Okay, thank you very much. I'm afraid we have to continue. Thank you, David. No.